So, bioinformatics. Let's talk introduction. Let's talk, what's going on, give you a little overview. So you can see a multiple sequence alignment going over our heads. Oh, we found a hit. So you basically break up a sequence into little bits, slide them along until you get the best match you possibly can. And when you've got that, then that is the alignment. And then you see whether you can align the things either side of it, and so on. And that's how programs like BLAST work, but we'll get into that. All right, first of all, here are a couple definitions of bioinformatics. Now, I don't think they're good definitions, but they are official definitions, so they count for that, right? So, application of computational tools, I agree, and approaches on molecular data, I agree, including the means to acquire, analyze, or visualize such data. It's okay, but it leaves out stats. You need to have statistical confidence tests at basically every stage of this in order for the results to have any meaning whatsoever. Because you and I can predict whatever we want, but that doesn't make it true. Yeah, it doesn't even make it relatively likely. There's no truth in bioinformatics. There are just good and bad predictions that usually do or don't pan out later. And everything in bioinformatics is destined to either be confirmed or refuted by actual bench experiments in the future, basically. Okay, so that was the NIH's version. This is the National Science Foundation. And so, oh my gosh, they have a lot to say. So, large databases. Okay. Genomes of organisms. Yeah. Molecular dynamics of proteins. That's like how proteins move. Population dynamics. How populations go up and down and what signals determine whether that happens. And basically, within all this area, is an enormous space. And so, bioinformatics is kind of the key. It's like how we unlock all this data. And by unlock, I mean we turn it into something that humans can actually interpret. Yeah. Okay, so bioinformatics is where multiple groups come together. You got your biologists that are collecting all this data because they want to answer some kind of question. It's now easy to collect data. It's easier to collect it than it is to analyze it, which is the big new shift in biology. But since you're still just learning biology, this is the way it is now. If anybody tells you otherwise, they're living in the past. Yeah, don't listen to fossils. Admire fossils, but don't listen to them. So you've got computer scientists that are actually building software, usually working with biologists that will actually help to analyze these things. And then bioinformaticians are people who can use the tools that computer scientists make on the data that biologists generate and actually answer important questions or make it interpretable in some way. Because, I mean, data is a bunch of ones and zeros, but they don't mean much to us. And yet, if you leverage it right, you can answer a lot of questions. So what do we actually do? For example, we'll take genes, we'll look for the open reading frames, which are the things that could possibly generate a protein. Once we've got a protein, we can look for functional sites, like is it the active site of an enzyme, something like that. And we can also try to model the structure of the protein or the structure of the DNA or RNA itself. And sometimes the structure will actually give you a pretty strong suggestion of what the function is. And so you can go from information to knowing what that actually does in a living organism if you do it right with reasonable statistical confidence. Yeah. Okay, another thing we do is assemble sequences. So you can generate a lot of sequence data, but it comes in itty bitty little parts. And so the secret to this is you have to find out where those itty bitty parts overlap. Like you see the little lines overlapping here, or here's an example. See how this part is the same as this part? They started from a sequence that's randomly shredded DNA, like shredding many, many copies of it, and some of the shreds, you hope, will overline with others. And then you want to sequence it so that you have 500 depth at every point, like 500 sequences giving you information at every nucleotide on the genome, or 1,000 or 10,000, whatever. The deeper your sequencing goes, the more secure you can be in the conclusion that these two bits actually do overlap. So this is called either third generation sequencing or next generation sequencing, and it's a big deal. There's a lot of money in it if you can get good enough at doing this. There's genome annotation. Once you put your genome together, what are the genes? What do they do? What do they tell you about the evolution of the organism that they come from? All great questions. Yeah, they can be answered with bioinformatics. At the level of looking at individual stretches of DNA, can you find the promoters that'll make the messenger RNA? Can you find the transcription factor binding sites? A transcription factor is a little protein, and it only looks for certain nucleotides to bind to. And if you have the right nucleotides in the right spot, it'll bind, and you'll get transcription just downstream. There you go. And then once you've got that, can you find the open reading frame? And so it's going to start with an ATG, like an AUG if we turn it into RNA, and continue all the way until it goes to a stop site. Yeah, you can find all these things with bioinformatics, and it turns just a nonsense of ACGT into something that's actually kind of useful. But it goes further. So all the genes inside your body are making proteins or making functional RNAs that are doing other things. They're interacting with each other. They're controlling processes. There are whole gangs of them that will be made in response to a particular kind of stimulus. And they'll all work together in some way, or they'll, work separately but to accomplish a common goal. You can arrange all these proteins and their functions in networks of genes that all get turned on together, networks of proteins that interact with each other and control each other. And if you understand that, you have a good idea of what the cell is actually doing. At least it's the best picture we can get at the moment. You can also study evolution as it is written in the DNA. By comparing genomes, you can compare open reading frames, identify versions of the gene that look similar and probably are homologs. They share common ancestry, or orthologs where they've been copied and you get multiple ones exist, paralogs, which are multiple copies in the same genome. Within those, you can find important sites within them. So you can figure out if we need to disable this enzyme, which parts should we target, or which are the important parts. The cool thing is that evolution will change anything that can be changed, given enough time. And if you stack up proteins and align them, and you find a part, like our little hit up there, that did not change when everything around it changes, you're probably looking at a very important part of the protein. Because probably it did change, but every time it changed, the organism died. And so it's an important part of an important protein, and that's why it will not show much variation. By comparing the version of a protein you get in several genomes, it's really useful for building up a hypothesis of what this gene does and how to figure it out, how to actually conduct the experiment that'll tell you exactly what it does and, turn it on and turn it off. You've got evolution, where, so this is the example from the 2009 pandemic flu, the swine flu, yeah, it's not really swine, but it's got elements, it's got segments that came from literal swine flu, like the kind that pigs get. But it's got elements, these two yellow lines, that came from a circulating H1N1 that humans get. And it's got elements that come from a different human infecting virus, the H3N2. And this mix of segments together was just the right set that just happened to set off this giant pandemic, which was H1N1 back in the day. There are people trying to do the same thing for SARS-CoV-2. They don't have quite as much data to work with, but you can still try it. And you can try and figure out, okay, we've got pressure coming from here. What are the different sequences that could have started this? How could they have recombined or not? How distant in time are these sequences from each other? Because most organisms, even viruses, will make a certain number of mutations in a certain amount of time. And so if you're, 30 mutations different, that's about 1 year of evolution for an RNA virus. And if you're 1,000 nucleotides different, that's a lot more years of evolution for an RNA virus. You can compare different hypotheses. So as we can make alignments, we can start to build family trees that will actually show the path of evolution that leads up to the things that exist now. Now, with these paths, we're going to be missing a lot of intermediates, all the things that happened and were not captured, but we can roughly reconstruct them, and we can do this reproducibly. It's really cool. And so you can compare different scenarios. So this is for SARS-CoV-2, the COVID virus. Did it go from bats to pangolins to humans? If it did, we would expect something like this. We would expect all the pangolin viruses to be here and the human viruses to be over here. Did it go from bats to pangolins multiple times and you had multiple entries in the human population? Well, then you'd have different pangolin strains that are closer to individual human strains. Yeah, possible, but that has been ruled out. That didn't happen. Or do you have, and this is the one, scenario 3, the first two were kind of straw men, I think, that actually seems to be the most likely, which is that pangolins get viruses from bats and people get viruses from bats. And the two are close, but they're not the step before. And the step before is one of these little nodes back here that we don't understand yet. It's a sequence we don't have that actually occurs in the past. And yeah, sometimes there's no way to get it. You can infer it, but you can't ever actually prove it. And sometimes with bioinformatics, you can actually go back and reconstruct what this thing could have been. Yeah, it's really neat. You can compare genomes at the chromosome level. So like comparing mouse chromosomes and human chromosomes, you find that we'll have similar numbers of chromosomes. But if you look at the colors, the regions in the human chromosomes have actually been broken up and redistributed as chunks. And that's what a mouse chromosome is. So the arrangement of chunks of genes within chromosomes does change over time. You get diseases where, for example, part of one chromosome will break off and attach itself to part of another chromosome. These are things that are still happening and mark some of the reasons behind evolutionary shifts. Because once you have a big break like that, if it affects the way the chromosomes pair up, basically you're a different species now, like it or not. And I hope there's somebody for you to breed with, because otherwise, yeah, too bad. You can find similar genes in very dissimilar animals. You can make alignments. Now look at this alignment of pre-pro insulin. So insulin gets cut twice. It gets cut, the pre-insulin, we cut off one little bit, which is at the beginning. And then pro-insulin, we cut out a bit in the middle and join these other two bits together. But you notice we're comparing Xenopus, which is a frog, and Bos taurus, which is a cow or a moo or whatever you ag people call them. And if you're looking at these, so there's a star underneath the alignment everywhere that they are identical. There are two dots wherever the amino acids are similar, and there's one dot where they're not very similar but a little bit close. And then there's just nothing where they're completely different. And you can see there are two chunks where you get a bunch of stars, from there to there and from there to there, where almost everything is conserved. Then you look at the region in between and boy, that's different. You've got deletions or insertions in there, you've got changes, it just is quite different. And you find out that aligns exactly with what the gene actually is and does. You've got an A chain at the end, a B chain in the middle, and the little C peptide is going to be cut out, as is the signal peptide at the beginning. The signal peptide changes a bunch because all it has to do is be cut by a particular enzyme called signal peptidase, I believe. The C peptide is going to be cut out by another enzyme, and as long as the cut sites on either side are fine, it doesn't really matter what the middle says, and it shows. And then the A and B chain are actually the insulin molecule that's going to go and bind to cells and regulate sugar uptake. And if those change, then the receptor has to change, and if the receptor changes, all the things the receptor interacts with have to change. And so that's a much bigger evolutionary step. And so, yeah, where do you see the changes? You see them in the signal, and you see them in the C peptide. Signal right there, C peptide right in the middle. Bam. You can't see on this one where they actually start, but by aligning them, you can kind of see where they start. Yeah, all right. Okay. And there are various elements in the human genome that are extremely conserved, like way more conserved than the things around them. These tend to be really important. They're going to be involved in things like RNA processing, like splicing, for example, or regulation of transcription. They can be transcription start sites and stop sites, or they're really important in development. Things like chromosome centromeres, very little change there. Because if you change one, you've got to change both at the same time, and the odds of that happening are very small. Because if you only change one, then they don't pair, and if they don't pair, they don't divide right, and then you just get dead cells, instead of children, basically. And just to wrap this up, so the other branch of this is going to be called functional genomics. You say, okay, what's the genome? What are the genes that come from the genome? When do they get turned on? How much do they get turned on? How do they interact with each other? And when they interact with each other, like if you have a regulator and an enzyme, does the regulator control the enzyme and actually turn it on and cause it to make more products? And you can actually measure the amount of product that gets made. That's called metabolomics. It's like looking at metabolites, little molecules that are produced. So there's like tiers. You've got the genome, you've got the, you can call it expressome if you're a weirdo, but I would call it a transcriptome. That's the more common term, which are the parts that are actually made. You can look at what proteins those actually produce. That's the proteome, all the proteins that can come from a genome. And then those are going to be, if they're enzymes, they're going to have particular products that they turn into particular, or reagents they turn into products. And so the connections between those are going to be the metabolome. And knowing all of this together builds a really deep picture of what's going on inside the cell. It's useful in cancer therapy. It's useful in understanding the world we live in, just in general. For example, in cancer therapy, you can say, okay, we've got these people who have this particular type of cancer. In this case, it didn't relapse. What genes were turned on or off? And so the genes that are turned on are going to show up as green. Genes that are turned off are going to show up as red, and everything in between is sort of yellowish, beigey, grayish colors. And so look at all these differences between the ones that relapsed and the ones that did not relapse. Look at that. You can see a very clear distinction between those. And so understanding which genes actually get activated or deactivated when a tumor metastasizes, starts to multiply, whatever. Very important. That's like the nuts and bolts of understanding what a tumor is and, therefore, how to fight it. So all these things come together. And the transcriptomes, the genomes, the epigenomes, which are little marks on the genome that control how it's actually expressed, the proteomes, the interactomes, interactions of different proteins, the networks, the metabolomes, they come together, and this is what gives you a phenotype. Together, these are the reasons why you're a little different from the person to the left and the person to the right. Together, these make you who you are. As we're all human, we're all mostly similar, but it's the little differences that add up in all these different ways, and that's what we actually see. So other interesting things, and this is one that is near and dear to me, structural genomes. We try and solve the actual shape of proteins so you can see them, and then you can figure out how they fit together, because they fit together like puzzle pieces, but first you've got to visualize, you basically got to solve what the puzzle is, what puzzle piece is, then you can figure out how to fit them together. Tricky. And so there's structural genomics, where the goal is to take every single gene, produce every single protein, and solve the structure of everything. Nobody's successful in this, but there are a lot of people trying, and they make a ton of progress. There are so many new structures coming out of these things. And that's, my background is I worked with one of these groups for a while. Very interesting stuff. And so, they're going to involve selection of targets, crystallography, actually solving these things. That's going to be fed by genome sequencing. It's going to come off in terms of characterization, like what do the proteins do? And once you understand the functions and their metabolic pathways, now we can basically go and look for those things happening in the cell. We can change the environment in the cell and see how that affects it. That's the idea. That's bioinformatics in a nutshell. It's figuring out all these things. So bioinformatics, not just one thing. It's like a whole toolbox. And it's got different tools, and each tool is real good at one or maybe two things. Like a hammer's awesome, but you don't want it to try and screw in nails. Or screw in screws, yeah. And a screwdriver is great, but it's not going to tighten a bolt. A ratchet is awesome at tightening certain kinds of bolts, but a ratchet is not going to plane a piece of wood down till it's flat. Not easily, not well. So yeah, different specialized tools that do different specialized jobs that together build a beautiful, beautiful construction. That's bioinformatics. That's the essence of it. All right, there you go.